Welcome to our Facebook Live, where UC Davis Mind Institute experts provide information about neurodevelopmental disabilities. I'm Marianne Sharp. Today, we'll be talking about the impact of chemicals in our environment on autism and neurodevelopment. We're joined by a real expert in this area, the Mind Institute's Pamela Line. Dr. Line is a professor of neurotoxicology and chair of molecular biosciences in the School of Veterinary Medicine. She studies the cellular and molecular mechanisms by which environmental factors, specifically chemical contaminants, modulate neurodevelopment and their possible role in autism. Thank you so much for joining us, Dr. Line.
Oh, thanks for having me.
Yes, it's a real pleasure. If you have questions, please post them in the comments now, and we will answer them toward the end of the discussion. And if something comes up as we're chatting, please post your question in the comments at any time. Okay, so our topic today, it's pretty wide-ranging and very complex. So I think we should start with a real big picture overview, and then maybe we can dig down more into some of the specifics, especially your work. So what do we know about the relationship between the chemicals in our environment, which many of us are exposed to all the time, and autism and neurodevelopmental changes?
Great question, and it is very complex. But the thing that I think we can summarize is that most people who work in the field of autism, clinicians, geneticists, as well as toxicologists, which I represent, are now coming to a consensus that the factors that drive individual risk for autism include not only genetic factors, which we've appreciated for a very long time, but also certain types of chemicals in our environment. And so what these chemicals appear to be doing, again, is modulating or determining our individual risk for diseases like autism. And so they don't necessarily cause autism. And in fact, I want to make that very clear that chemicals themselves are not causing autism, but what they are doing are interacting with these genetic risk factors to modify individual risk. And sometimes that modification may be to decrease risk, and sometimes it may be to increase risk.
What was the genesis of this field of study? How did researchers first figure out that something was going on here that we needed to look into?
Yeah, that's a really good question too. And I thinking about this, there are really three independent lines of evidence that got us to this point today. One of those is the very alarming increase in autism over a very short period of time. So if we look back over time, the number of kids that were being diagnosed with autism, 50 years ago was significantly lower than it is today. And so this first really began to raise alarm bells for toxicologists that there might be environmental factors that are really driving this increase in the incidence of autism. There have been other alternative explanations for the increase, such as diagnostic substitution, which basically means that the diagnostic criteria for autism has evolved. And so that kids who may not have been diagnosed as autism previously under the former criteria are now being diagnosed as autism. And certainly that has contributed to this apparent increase. But a number of studies have very clearly shown now in different areas around the world that there is a real increase in autism. And the rate over which that autism has increased is too rapid to be explained by evolutionary drifts in human genetics. And so the other possible explanation, of course, is that we have factors in the environment that have changed over that time frame that are contributing to this increase in autism. So that's line number one, and a very compelling line of evidence. The second line of evidence are the genetic studies themselves. Once it became apparent that autism was increasing, of course, large numbers of dollars and research effort were pumped into looking for genes that were driving this risk. And what the genetic studies told us, number one, genes are very important, but number two, genes aren't the whole story. So there have been very few single gene mutations that have been linked to autism, and those that have represent an extremely small percentage of the cases of autism. Rather, it's a much more complex genetic picture. We even know that people who have the same genetic background, which confer risk or increase risk for autism, don't show the same clinical phenotypes. Again, suggesting there's some factor in their environment that's interacting with those genes to really determine what happens clinically. So that was line of evidence number 2. And then the third line of evidence is back in the 1950s or so, there began to emerge very strong epidemiological and clinical evidence indicating that congenital rubella, so in other words, when pregnant women got rubella, which is a type of viral infection, their children tended to have significantly increased risk for autism. And also women who used valproic acid, which is an anti-seizure medication, also had an significantly increased risk for having a child later diagnosed with autism. And so this is another compelling line of evidence that kind of told us that there are factors in the environment that are going to be influencing risk for autism. So those 3 lines of evidence all kind of came about at the same time. And people putting them together said, wow, we need to be looking at the environment because we can control the environment to some extent. We can't currently readily control genes, but we can control the environment. So really important to identify what those factors are.
Yeah, well, as you just mentioned, it's more the interplay likely between genes and environmental factors that might be associated with autism. But can you explain what researchers think is actually happening there? I'm sure that's a really difficult question, but what are the theories?
Yeah, so this is a very hot area of research, and it's a rather controversial area of research, as you can imagine. So the 2 outstanding questions are, what are the environmental factors that are driving autism? Which genes are they interacting with? Because clearly the evidence would tell us that the chemicals themselves are not causing autism. So what are the specific genes interacting with specific environmental factors that are driving risk for autism? And then secondly, how are they interacting to increase risk? And that second question is pretty murky, I'm going to be honest. And so the hypotheses that are out there, there's a handful that are being actively explored. Probably the one for which there's the most evidence or the strongest evidence is that environmental factors are modulating or changing the same signaling pathways in the developing brain that are changed by the genetic mutations. And so because they are acting on the same signaling pathway, when you add them together, that's amplifying the effect. And so with anything in biology, we have a gradation in response, right? So just the genetic factor itself, all by itself, will increase risk a certain amount, but maybe not over a threshold to actually result in clinical manifestation of autism. But if we have 2 genes that converge on that pathway, we raise the threshold or we raise the risk, and it might cross that threshold. Or if we have the genetic risk factor and an environmental factor that impinge on that same signaling pathway, together they increase the risk above the threshold. And so that is one explanation for how environmental factors may be interacting with genetic factors. And I think, again, we have the strongest evidence to support that. Another possibility is that environmental factors, and particularly chemicals, are metabolized by our body. So when they enter our body, they get changed, physically changed or chemically changed from the parent compound, as it's called. Enzymes act on that to metabolize it into a product, which is slightly different from the parent compound. This system has evolved over time in our bodies, this enzymatic system, to detoxify chemicals or to make them less toxic. In some cases, though, they actually make the chemical more toxic, so they're toxifying. But what's been proposed is that, and this is based on some evidence, that some of these autism risk genes are actually changing the activity of these enzymes that are metabolizing the chemicals. So they're changing the activity of the chemical. Again, sometimes decreasing the toxicity of the chemical, sometimes increasing. And so that's another really strong hypothesis that's out there for how chemicals and genes interact. And then there's a bunch of other stuff, which is definitely still in the exploratory stage, but there's suggestions that perhaps chemicals and autism risk genes act on the gut microbiome together. We now have a lot of evidence that the gut microbiome influences the developing brain, and so those effects may amplify to drive increased risk. There's some suggestions that perhaps chemicals are interacting on the immune system to cause immune changes that are associated with increased risk. And of course, if you have genes that are also changing the immune system in that direction, they can amplify each other. So in a big picture sense, really the thought is that somehow these are interacting, that they amplify, the genes and the environment are amplifying each other's adverse effects to push the individual over that threshold for manifesting clinical phenotypes.
Wow, that was a great overview. So many questions, right, still to answer.
Yes,
Uh,
there is.
and you've done a lot of very specific research focused on PCBs or polychlorinated biphenyls. I hope I said that one right.
You did.
So these are those man-made chemicals, and they're really hard to avoid in our environment. So could you maybe first talk about what they are and what you're finding about PCBs and their link to possible link to autism?
Yeah, so PCBs, as you indicated, are man-made chemicals. They were synthesized in very high volumes from about the 1920s to the 1970s. They're a relatively simple structure. So they're two, if any of you remember your introductory chemistry, they're two phenyl rings, so two rings of carbon that are attached to each other. And where hydrogen would normally be off the carbons, there are various chlorines attached to them. And you can imagine there can be differing numbering of chlorines in different positions on those rings. So there's 209 possible versions of PCBs, which has made this kind of tricky to study. But nonetheless, these chemicals were made in such high volumes because they're incredibly stable and they are really good at dissipating heat. And so they were put into hydraulic fluids to keep the hydraulic fluid from heating up too much. They were put into the ballast of fluorescent lights to keep the lights from overheating. They were put into electrical transformers. So they're in the oil and electrical transformers, again, to dissipate heat. But we also find them in caulking material, in paints, in adhesives, where they are playing a role in not only dissipating heat in some cases, but also they're very plastic. And so they increase the durability and sort of flexibility, if you will, of all these materials. And so they were in a large number of industrial and consumer products. It wasn't till the 1970s that people began to be really alarmed about the increasing presence of PCBs in our environment. Because they are so stable, they don't break down very quickly. Our bodies cannot metabolize them very quickly. And they tend to be lipophilic, which is a scientific term, meaning they like fat. And so when they get into our bodies, they are stored in our fat tissues, in our adipose tissues. And unfortunately, the brain has a lot of materials that look like fat. It's the myelin sheaths that surround the nerves. And so we have a lot of PCBs that are stored in our brains as well. But in the early 1970s, there was an alarming series of studies coming out of labs around the world saying, hey, these PCBs are not breaking down, their levels are building up in the environment. And number two, we have very strong evidence suggesting that at least a subset of these PCBs are increasing risk for cancer. And so based on that information, the United States Congress actually passed legislation banning further production of PCBs in the United States. And then subsequently, back in the early 2000s, there's a group known as the Stockholm Convention that basically banned production of PCBs worldwide. And so PCBs are no longer produced intentionally, but they're still produced unintentionally as byproducts in particularly commercial pigment synthesis. And so we still find these PCBs being produced today, again, not intentionally, but sort of unintentionally. And so, you know, the critical things to remember is these are a type of forever chemical. You've probably heard that word in the news. It's been in the news quite a lot in the context of another group of chemicals known as PFAS. But PCBs were sort of the original forever chemicals, and they're around forever. And we now know that not only do they cause cancer, but another different subgroup from those that cause cancer also can interfere with the development of the brain.
So what is your research zeroed in on? What are you finding regarding PCBs and autism?
Yeah, so we got involved in PCBs when I first started my research career because I was very interested in chemical interference with brain development. And PCBs were well appreciated at that time through a series of beautiful and elegant epidemiological studies to cause neurotoxicity in kids. And so we wanted to understand how they were doing that. So what was the mechanism by which PCBs changed brain development? And what we eventually stumbled upon is that PCBs are very sneaky. So they're not causing frank toxicity in the brain. They're not killing brain cells, but what they're doing is they're getting into the brain and they're turning on the same signaling pathways that control how neurons develop their shape. And so neurons are the cells in the brain that are responsible for transmission of information. And neurons have very, very specific and complex structures. So they have a cell body like all cells, but then these processes that extend off the cell body. And these processes are just like wiring in a computer. They connect up with each other to form circuits through which information flows in our brain and then into the periphery, into our sensory organs, into our muscles, et cetera. And so these get laid down during development in a very precise way to form functional neural circuits. And if you change the way the neurons develop, so you change the number of extensions they have, or their length, or their shape, you change the circuits. And sometimes these changes can have functional consequences. And so what we found is that PCBs were changing the structure of neurons early in development by changing the signaling pathway. So they specifically turned on the release of calcium, which is a signaling molecule. They increased calcium levels in the cell body, which then triggered these signaling pathways that increased process extension during development. So they were hijacking the normal signaling pathways that would control extension of these processes from the neurons during development and causing it to occur inappropriately during the development of the brain. We later found out, so we did all that research in PCBs for about 10 years, and then I got interested in autism, and I find out that the exact same signaling pathways are turned on by certain autism risk genes. And so it turns out there's a very large category of risk genes for autism that control calcium signaling. And so they're basically turning on, as I said, the exact same pathway as the PCBs. So then we start looking at the interaction between these two.
Wow. So if we could just dig a little more into how this sort of research is conducted. So what are the best ways for researchers to study this interplay, right, between chemicals in the environment and our genes? Is it animal models, natural exposure, behavioral studies, or is it all the above and more?
It is all of the above, so each type of study has strengths and it has weaknesses. And so, I mean, my work has really very much focused on animal studies and cell culture studies, but I will start by saying epidemiology is actually critically important because it was epidemiologic studies, so studies of humans, that really identified that PCBs were neurotoxic to the developing human brain. But to really understand how they're causing these effects, you really do need to have animal studies, and these include behavior studies. And then you also have to have cell culture studies where you can really get in and manipulate the molecules inside the cell to see what's really driving these changes in cell shape. And then how do these changes in cell shape relate to the behavior, the effects of PCBs on the behavior? And so we've done a very careful study over the years to look at everything from the animal, the behavior, look at the structure of the neurons in the animal's brain after being exposed during development to PCBs, using exposure routes that humans would be exposed to PCBs, and then taking that into the dish and saying, we get the same changes in the cell in the dish, and now let's start playing around with the molecules using our various tools that we have to change expression of molecules in the cell to see if that changes the response to the PCBs. So yeah, all these are very important types of studies.
If you're just joining us, we are talking today about chemicals in our environment and possible links to neurodevelopment and autism with the Mind Institute's Pamela Line, a professor of neurotoxicology and chair of molecular biosciences in the School of Veterinary Medicine. If you have a question for Dr. Line, please do leave it in the comments and we will get to it toward the end of our discussion. Okay, we talked about PCBs, a big area of focus for you. And you alluded to one other type of chemical, but I wanted to broaden the discussion. What other chemicals have been linked through research to neurodevelopmental changes? And lots of things come to mind like pollution and pesticides, et cetera. It's a long list.
It is actually a long list and a growing list. So if we focus on just neurodevelopment and not autism specifically, the list is much longer. And so there are a couple of compounds that I'm sure many people in the audience are very familiar with as being neurotoxic to the developing human brain. And that includes things like lead, methylmercury, certainly pesticides, and there's many different types of pesticides. One class that has been very strongly linked to neurotoxicity in the developing brain are the organophosphate pesticides, such as chlorpyrifos, which has been notoriously in the news. We also know now that air pollution drives adverse changes in the brain. And of the group I've so far mentioned, the organophosphate pesticides and traffic-related air pollution specifically have been tied to increased risk for autism. Other chemicals we're very concerned about are the brominated flame retardants. We also are concerned about the new forever chemicals everybody's talking about, the PFAS and PFOS chemicals. So yeah, there's quite a broad range, but in general, many of the heavy metals, many compounds that have halogens on them, like chlorine or bromine, we tend to really be concerned about in the context of brain development.
Yeah, I mean, I have to say a lot of what you're talking about, we're all exposed to some of these things, most of these things at one point or another. So how do researchers pinpoint why certain people go on to develop changes or autism or have neurodevelopmental changes of some kind, and then others do not? Is it just the genetic factor? Is there more to it?
Yeah, there is more to it. We've already talked about genes at length, and that's probably one of the more critical factors. But other factors that are really important depend on the exposure. And so when we talk about exposure, there's two factors that are really important. One is the timing of the exposure relative to where you are in your life. So for many of these chemicals, the mature brain or adult brain is relatively resistant to the neurotoxic effects, whereas the developing brain is much more vulnerable. There are exceptions to that, of course, as with biology, there's exceptions to everything, but in general, the developing brain is more sensitive to chemical insult. So the timing of exposure is a really critical factor. And even within the developing brain, timing can be very important. So depending on what the target is for that chemical, exposure very early in development might have no effect, whereas an exposure later in development might, or vice versa. So it's very complex, but exposure timing is really critical. The other one, of course, is the level of exposure or the dose. So almost all chemicals, there are exceptions again, but almost all chemicals have a threshold. So below that threshold dose, you're unlikely to see an effect. Above that threshold dose, you have an increasing probability of seeing an adverse effect. And so timing, dose, genetics are all the really big factors that are going to be important in determining whether somebody has an adverse effect or not.
Yeah, that makes you think about the role that location might have. You live near a highway, maybe you're more exposed to auto emissions. We have people who work in the fields, maybe more exposed to pesticides. Is there research looking into certain location-based changes?
Absolutely. Yeah, absolutely. I mean, that of course determines your exposure, which you just said was a very important
Yeah.
factor in terms of determining outcome. And so this is actually a tool that is used by epidemiologists to have an location where there's a heavy exposure and to compare outcomes in that population versus a population that has low level exposure. But it also pinpoints, I think, why it's so challenging to identify environmental risk factors that are driving risk for autism, because there's all these variables. And so it's people will say, well, study X found a link, but study Y did not. So what do we make with that information? But what we make with that information is that's kind of to be expected. But the people in study X just have other factors that are increasing their risk for an adverse effect related to that exposure.
And is there anyone looking at, say, certain combinations of different types of chemicals or total
Yeah.
impact? Is that just too far down the road?
No, actually, those types of studies are underway. As you can imagine, they're very expensive studies. They're very complex studies. We've been engaged in, actually, before I get into what we were doing, I just should mention quickly for the audience that there's actually a very large research initiative in this country to look at something referred to as the neuro-exposome. And that term exposome is meant to be a collective term that refers to our collective exposures. And in the context of chemicals, that's not just environmental contaminants, but also drugs that we're putting into our bodies, foods that we're putting into our bodies, consumer products that expose us to chemicals. So all these factors contribute to the neuro-exposome. And there is, as I said, a new research initiative in this country to try and understand A, what that is, and B, how that relates to disease. But in my own lab, our attempt at this is I had alluded to the fact that there are 209 PCB congeners. And so we're not exposed to a single PCB, none of us. I could take a sample from all of us in this audience and I'd find multiple PCBs in all of us, although the profile would vary between all of us because we've been in different locations, we have different enzymes, we have different dietary factors. So, it's going to be slightly different between all of us. But what we've done is team up with other research and scientists at the Mind Institute to leverage an ongoing study at the Mind Institute called the Marble study. And so the Marble study is recruiting women who are increased risk for having a child with autism. And these are predominantly women who have had one child already diagnosed with autism, which means they have an increased likelihood of having a second or third child that also has autism. And so these women have been recruited at the time of pregnancy. Samples have been taken from them. They've had survey data collected from them to try and determine what their exposures to chemicals have been during gestation and maybe prior to gestation and after gestation. And so we have a pretty good profile of PCBs that have been detected in these women. And so in the laboratory, we took that information and made up a synthetic mixture of PCBs we called the Marbles mix. And so this reflects the 12 most abundant PCBs that were found in the Marbles moms during pregnancy. And we've used that to administer to our animal subjects and also to study in culture to say, okay, here's a human relevant mixture as opposed to looking at a single PCB, which we did in our early studies. Do we still see the same effect? And the answer is we do. But the dose response curve is shifted a little bit because the ratios of these chemicals are slightly different. There are other people who are doing similar things by looking, for example, at populations that are increased risk for autism and saying, okay, their primary protein source, which is the primary source of exposure to PCBs, are fish from the Great Lakes. So what do we find in fish from the Great Lakes? And they're trying to recapitulate that chemical mixture and then use it in their experimental studies.
And we will put a link to the Marbles study in the comments, as well as a link to Dr. Line's lab webpage, which has a whole host of links to studies that papers she's written, as well as other documents that should be really interesting. In fact, apparently the link is already there. So you can check that out now. Okay, Dr. Line, in addition to focusing on autism and PCBs, you also study the impact of genes and the environment on some neurodegenerative diseases such as Alzheimer's, and you also study asthma. So maybe could you give us some insights into what you've learned about environmental links between Alzheimer's and asthma?
Sure. So the asthma studies, and you're probably thinking, why is a neurotoxicologist studying asthma, which is a disease of the respiratory system? But it turns out that in humans with asthma, as well as in animal models of asthma, that it's the nerves that innervate or control the airway smooth muscle that are affected in this disease. So they are typically dysfunctional. And what I have found in collaboration with a colleague up at Oregon Health and Science University, Dr. Allison Fryer, is that organophosphate pesticides can precipitate or trigger an asthmatic attack and increase risk for asthma by changing those nerves that enter this airway smooth muscle. So they basically remove the inhibitory drive or the system that would normally prevent the nerves from becoming overactivated and then causing too much constriction of the airway muscle. That inhibitory control is removed by the organophosphate pesticides so that now when the nerves are activated, there's not a turnoff signal anymore. And as a result, we have overcontraction of the airway smooth muscle, which narrows the diameter of the airway, which of course is the classical symptom that causes of asthma. So what's really kind of scary from our research, but it's actually resulted in some regulatory change, is that this is caused by levels of organophosphate pesticides that would be considered safe under the previous guidelines. And so typically we have regulated these pesticides based on whether or not they're inhibiting an enzyme that we can measure in the blood. This enzyme is acetylcholinesterase. So for example, pesticide applicators or people who are working with pesticides in factories would be tested routinely for the levels of this enzyme in their blood. And if it wasn't inhibited, they say, we're good. But we now know that these levels that don't inhibit that enzyme in the blood will trigger asthma. So obviously, this was kind of a big revelation. In terms of Alzheimer's disease, our work has very much focused on air pollution. When we look at the epidemiological data out there on Alzheimer's disease, we see that the environmental factor that's most strongly associated with increased risk of Alzheimer's disease is air pollution, and in particular, traffic-related air pollution. So the air pollution generated by cars, trucks, et cetera. And so while the epidemiological data is very strong, we of course need animal models to corroborate that, because there's a lot of ways that we could have this association, and it has nothing to do with air pollution. You know, people who live near roadways tend to have a lower socioeconomic status, et cetera. So we developed this really pretty interesting model, I think, where we said, okay, we don't want to collect air pollution and bring it back to the lab, because in a collection, we're changing the characteristics of the air pollution itself, we're concentrating it, and we're not replicating the diurnal or daily rhythms we see in air pollution or the seasonal rhythms that we see in air pollution. So we decided to build a vivarium, which is basically housing for rats, a vivarium next to a heavily trafficked tunnel here in the Northern California area. So we call this our TRAP project, for traffic-related air pollution. So basically we park these animals for up to 14 months of their life next to this tunnel, collected air from the tunnel, and delivered it to the animals in real time, unchanged. So the animals are breathing exactly what people breathe, who are commuting or who are living or going to school near a roadway. Our control animals, who were housed in the same vivarium, were exposed to filtered air. So we collected air from around the trailer, which would have been polluted because there's lots of cars there. And we put it through a series of filters to remove all the gases and all the particulate matter, which are thought to drive a lot of the toxic effects of air pollution. And what we found is that indeed, the animals that were exposed to the trap had a much higher incidence or more severe manifestation of Alzheimer's disease-like pathology in their brains, and they had behavioral deficits, and these occurred much earlier in life than they did in the animals that were exposed to filtered air. So now we're doing studies to try and figure out how it is. Well, two studies. One is to figure out which component air pollution, the particulate matter or the gases, or a combination, are driving this increase in Alzheimer's disease-like phenotypes. And then secondly, what is the mechanism? So what is TRAP doing inside our bodies that actually is increasing the expression of these Alzheimer's disease-like symptoms?
Yeah, well, that leads really great into the next question, which was, what are the approaches that individuals can take to minimize exposure to environmental chemicals? You know, you're talking about filters, just, you know, is there anything that you do or that you would recommend that people do?
Yeah, there's actually quite a few steps that we can do to sort of minimize our exposures. None of us are ever going to have zero exposures. It's not possible. But certainly we can minimize our exposures to a number of things. And so I think it's probably hard for the layperson. I have a bit of an advantage because of my training to know which chemicals are going to be protected by any specific activity or action that you may take. But certainly with regards to air pollution, I think it's pretty straightforward. Is you can definitely pay attention to the air alerts. I do this, and on very high pollution days, I definitely wear a mask when I go outside, and I limit my time of doing vigorous exercise outside during those high air pollution days. And these are very straightforward steps that I think most people can take. Definitely keep your windows closed. If you can afford an air purifier in your home, they're definitely worth the investment. And of course, if you can avoid heavily trafficked areas, choose routes that don't take you through those areas if possible. We all need to be using mass transit to the largest extent that we can, and avoid individually driving, etc. In terms of some of these forever chemicals, which tend to be fat loving, as I said, so they tend to accumulate in things that have fat. And of course, if we think about our diets, we have a lot of sources of fat in our diets. Look at fatty fish, look at red meats, which have a high level of fat. And so one thing that you can do is try and decrease your fat intake, cut away the fatty areas of these meats, or minimize your intakes of these types of meaty tissues. Cheese and dairy products also can have high levels of PCBs. So while there are health benefits associated with it, you might want to balance how much of these you're consuming. The other thing I think is a really interesting one is we now have very strong evidence that some of these PCBs that are unintentional byproducts of pigment production are released from gases and from caulking materials. And so we see very high levels of these in homes that have been recently renovated. So another approach that we can all take is, if we're in the process of renovating and using varnishes or stains or caulks or paints, is that we use very good ventilation in our homes, and if at all possible, avoid being in those homes during the time of renovation, or at least close them off and use ventilation in the other rooms where you're living. The thing that's really interesting for parents to consider, many of us will put together a beautiful nursery to receive our newborn when they come home from the hospital, and bright colors are often chosen. Well, unfortunately, it turns out that these really bright colors are the paint colors that tend to have the highest levels of these compounds. And so I would urge most parents to think about using soft tones for their paint walls, or neutral tones. So soft grays, soft whites, soft yellows. Don't go for those really bright sort of neon effects, because those tend to have the higher levels of these PCBs. And then in terms of pesticides, we actually have a new regulation that's being passed here in the state of California about pesticide notification. So notifying the public when public sprayings will occur. If you have such information available to you, please utilize it. Plan to avoid areas that have been recently sprayed. Make sure your windows are closed on those days. If you need to be in those areas, wear a mask. So there are some straightforward things that we can all do to minimize. Eat a balanced diet, get plenty of fluid, and sleep well. It's amazing how lack of sleep can increase your susceptibility to toxic chemicals. So.
All great advice. I mean, it can be a little scary to think about all of this, but as you've said, not everyone, I mean, we're all exposed to many of these things on a regular basis, and not everyone goes on to develop Alzheimer's or
Right.
asthma, et cetera. So as you talked about, that's really exposure levels, genetic background. Is there anything else we should, say to people that don't panic,
I think that's the key. Don't panic. However, I do think it's important to be informed. So many of our public health agencies do provide information, particularly to individuals who are planning to have a child or who are pregnant, about certain things you really should avoid during pregnancy to, again, minimize your exposure of your developing child to these chemicals. And so it's make yourself aware of that. And if you have questions, reach out to people who can explain it to you. It is important to be informed.
And then what are the implications of research like yours? In fact, you sort of alluded or mentioned to a couple, which is changes in regulations.
Yeah.
What are the goals behind arming ourselves with this information?
So the key goal is to really change regulation to protect people from exposures. And as you can imagine, many of these chemicals over the years have been very desirable commercially. And so in order to enforce changes at the regulatory level, it's really important to have compelling scientific data. And so this is really the goal of a lot of the work that drives me to do the work that I do, and many of my colleagues, is to provide that evidence. I mean, if a chemical is safe, we want to know that too. We want to know which chemicals are safe to use, because we will not ever live in a world, or it seems unlikely, at least in my lifetime, we'll live in a world where there are not chemicals. Chemicals are important for safety and for health as well. So they do have their benefits as well. So we want to know what are the bad actors, and that's part of our job as toxicologists, is to identify the really bad actors. And the second part is to identify how much of that chemical is dangerous. And so we use that information to provide to regulatory agencies so they can make scientifically rational decisions. And of course, regulatory policies require money to enforce, to enact. And so they need to have this really strong data to justify changing regulatory levels.
Okay, well, this has been really enlightening. Thank you so much, Dr. Line, for being with us today, for sharing your time, and of course, your expertise.
You're more than welcome. It was a pleasure.
And I encourage you to share this post if you have family members or friends that you think would enjoy learning from it. It will also be posted to our YouTube channel where it can be viewed indefinitely. And thank you so much for joining us for this UC Davis Mind Institute Facebook Live.
The UC Davis Mind Institute was founded in 1998 with a promise to reduce and prevent the disabilities that can be associated with autism and other neurodevelopmental conditions. Every day, our clinicians and researchers make progress on that promise. Our groundbreaking research on autism, Fragile X syndrome, chromosome 22q11.2 deletion syndrome, ADHD, and other conditions associated with disability are helping affected individuals achieve their fullest potential. Please visit our website or our social media platforms to find out more about current studies, upcoming events, and how you can help make a difference.